Cool. Thank you, Sarah. Hi, everyone, and thanks for joining us. I'm Saba Farnaghi, and the product manager for antibody portfolio at Joma Life Research. To assist you in cutting through the noise about what COVID-19 research products are available, we've been running a webinar series giving a 10-minute overview of the most reliable products. Today, I'll be talking about the use of GenScrape qRT-PCR kit for detection of SARS-CoV-2. While I'm presenting, our team of fantastic product managers will be available to assist. So if you have any questions during the presentation, please click on the Q&A button and it will open a dialog box, as Sarah mentioned. But also stay on the line following the webinar if there are any other issues you'd like help with. Well, what we know about GenScrape qRT-PCR kit. First of all, I would like to mention that GenScrape is the world leader in biotechnology reagent services. They offer complete assay with primers, probes, reagents, and positive control plasmid. This kit has no background noise, and is highly sensitive enough to detect 0.001 picogram of control plasmid. All the primers and probes in this kit were purified at the highest HPLC grade with more than 90% purity and less than 0.05% cross-contamination, leading to ultra-low false positive rate. GenScrape is a reliable supplier. They have low history of quality issues with a large capacity to fulfill your needs. They will also prioritize the production of any reagent related to COVID-19 study. Well, I would like to take you through the principle of the gold standard detection method for novel coronavirus. There are 4 steps. In step 1, we would like to isolate the novel coronavirus from the patient's and sequence its genome. In step 2, we need to compare the genome sequence of the virus with human genome in order to find out the specific sequences in the virus genome. At step 3, companies like GenScrape design PCR amplification primers and fluorescence probes for detecting those specific sequences. At step 4 is our role as scientists to perform RT-PCR. I'll be talking in more details about step 3 and 4 in a second. How did GenScrape design the primers? In late January 2020, NGS sequence data reported that entire SARS-CoV-2 genome is about 30,000 base pair in length and encodes 10 different proteins. Although 60 to 90% of the genome sequence is highly conservative and resembles other coronaviruses, scientists have identified a highly specific region, ORF1ab, RDRP, which is a part of ORF1ab gene. E and N genes are those specific regions. These genes share no common sequences with all other known coronaviruses, such as SARS and MERS. WHO and CDC later published a qPCR protocol against these 4 genes as guideline for SARS-CoV detection. According to CDC, the sample is determined as positive when ORF1ab and N genes are detected, while according to WHO, samples are considered positive when both R and genes are detected. We can see the sequences of all 4 genes and probes designed by GenScrape in this table. GenScrape also offers other customized primers and probes for your COVID-19 research. SARS-CoV-2 nucleic acid detection workflow typically includes sample collection from upper or lower respiratory tract, blood, stool, or conjunctiva, specimen if patient has eye infection. You can extract the RNA using a traditional triazole-based protocol or commercially available RNA extraction kit. Then you perform PCR and evaluate your result, right? GenScrape kit is compatible with many qPCR instruments, such as ABI PCR system, Roche, and Bio series of PCR systems. And GenScript kit typically takes 1 hour after RNA extraction. And also I would like to mention that this qPCR assay is based on TaqMan probe technology, opposed cybergreen qPCR technology. The mechanism is very simple. Through addition of fluorescence probe to the reaction. Due to time limitation, I'm not going into the details of probe-based technique today. For one-step PCR, you typically need to add reverse transcriptase enzyme, your template RNA, buffer, DNTPs, primers and probe, as well as DNA polymerase, such as Taq polymerase. When you add all the reagents, you have to seal the PCR tube and place it into real-time PCR machine and follow the protocol provided in the kit. GenScript offers one-step 1-plex and one-step 2-plex kits. The components of both kits are similar. You get master mix, enzyme mix, reference dye, if your instrument requires primers and probes, positive control, and RNase-free water. In 1-plex detecting assay, you have choice of 4 targets, and all fluorescence probes are detected by FAM channel, while in 2-plex kit, you can choose one of the gene pair recommended by WHO or CDC, and of course, you need to detect fluorescence probes by different channels. We're looking at validation data provided by GenScript. The PCR result should be or will be analyzed based on the number of PCR cycle before the fluorescence signal passes the threshold called CT value. A reliable qPCR assay usually has a CT value more than 40 for negative controls, and positive control CT value is around 26. The result then can consider positive if the CT value is between 30 to 37. As you can see on the left panel, all positive controls for 4 genes had CT value around 26, and non-template controls showed no signal for 1-plex assay. In the left side hand, for 2-plex assay, you can see similar result for gene pair, and negative control had no signal. So you've got your result. It's time to analyze your result. Like any other experiment, you need to set up negative and positive controls and meet expected result for each. Failure to obtain quality control indicates a failure of the test. You need to retest your sample or test in an alternative method. How to interpret is easy, as I mentioned. You consider your sample positive when the CT value is equal or less than 37. And you typically have the S-shaped curve, including a log phase. And if your sample CT value is more than 40 or you have no signal, then your sample or your result considered as negative. In a nutshell, if you need to detect SARS-CoV-2 in vitro, GenScript offers several detection solutions. Molecular diagnostic reagent, which I mainly talked about today, includes your choice of primers, probes, plasmids, or a complete qPCR kit. But if you're looking for serological testing reagent, GenScript also offers a wide range of proteins, antibodies, as well as ELISA kits. Now, we do have full list of GenScript COVID-19 product on our website. If you're interested in any of this item, please check it out. Lastly, I would like to let you know that we created T-shirt illustrated here in appreciation of your fantastic work during this pandemic. You can have one of these entirely free. Just email us your details and we'll send you one. Jomer is the exclusive distributor for GenScript in Australia and New Zealand. If you need a code or for more information, don't hesitate to contact us. That's it. Thank you for this afternoon webinars. We are bringing a poll that Sarah is shortly talking about.
Thank you very much, Saba. Fantastic presentation. Do we have any audience questions? Saba is here, and we are all here if you have anything that's on top of your mind. And just remember, no question is silly, and I'm sure that other attendees, it would help them as well. So while we're thinking of a question, I'm going to launch the poll, so that will appear in front of your screen. If you could complete this poll for us, it will help us create better content and new webinars in the future. So that would be fantastic if you complete that poll for us. Thank you. Great. Well, so Saba, I'll ask a question on, you know, just thinking about stuff. When can you purchase the kit components individually, or does GenScript offer the full package?
As I mentioned in my one of my last slides, you can purchase primers, probes, or plasmid separately, or you can choose to purchase qRT-PCR kit as a complete assay.
Yeah.
Some of the customers are not very familiar with the PCR method, which is also a very simple technology, but they can just go for very simple assay, complete assay kit.
Mm-hmm. Great, thank you. So Bin, if you're still here, Bin Ma, you had your hand up. If you have a question for us, please let me know. If you're still here, it appears you may have ducked away. Bin will get back to you via email with a follow-up after this seminar. Any other questions, feel free to email us at info@jlresearch.com.au. Now, this webinar will appear on our YouTube channel tomorrow, and we hope that you can watch the replay there. So thank you very much, Saba.
Thank you.
And thank you for everyone for attending.